Okay, we are back for part two, and just a bit of comment on how this thing has been going so far. And I have to admit that this is so much easier than what I have done previously, precisely because I have relegated to this laxed state of mind currently, and it helps me be free, and it's just like a very normal study session without any extra hassle of being too professional or being or putting any extra strain on me, because the core object should be learning this for myself. And any ounce of being entertainment or being informative stuff like that should become secondary, because this is how I conceptualized this routine for me, or like this act. It would consist of just have you seen the study with me videos? Someone is studying and you're like they're counting down to something, etc. So this is more like that. This is not supposed to be educative, this particular set of contents. So it helps me just focus on what is ahead of me, in front of me, and just study it out, read it out. Okay, can you explain to me the difference between fibrous? Okay, we were here trying to understand the difference probably between fibrous protein and proteins and integral and peripheral proteins. Elongated, strand like, sheet like, etc. We have done that. Relevance to cell membrane. While fibrous proteins can be associated with the cell membrane, proteins of cytoskeleton that attach to the membrane for structural support, they are not typically embedded within the membrane itself. Okay, I thought they were though. I thought the this particular one, this one, it kind of shows that fibrous proteins are embedded within the cell structure. But we are not going to quarrel about that, and we're quarrel about it. We are just going to move on. In the way integral proteins are, instead they usually form networks underneath the membrane skeleton proteins, or go outside the cell, extracellular matrix proteins. So I think what I am going to do is, which one is, yes, I'm going to leave it up to this. So I think this would eventually answer this particular question or solve this conundrum. Integral membrane proteins, a classification based on membrane association. Definition: these proteins are permanently associated with the cell membrane. They are embedded within the lipid bilayer, meaning they have hydrophobic regions that interact directly with the hydrophobic tails of the phospholipids. Again, these are more detailed elements that I'm going to read. I'm going to leave to the formal textbook on this. How they are removed: you cannot remove them simply by changing salt concentration or pH. You need to disrupt the lipid bilayer itself, typically using detergents which act like soap, breaking up the fatty membrane. Okay, so this was my remark here. My remark was like, is this why it is so slimy after you have eaten beef curry and stuff like that? The oily substance is this. Types: transmembrane proteins. Again, transmembrane proteins are the stuff that I have to understand in much more detail, and I'm going to leave it to the textbook here, this one. Many receptor, okay, these span the entire lipid bilayer, which parts exposed on both sides, and which both the inside and the outside of the cell. Many receptors, channels, and pumps are transmembrane. Integral monotopic proteins. These are embedded in only one leaflet, half of the lipid bilayer, and do not span the entire membrane. Function: these are workhorses of the membrane, involved in transport, signal reception, cell adhesion, and enzymatic activity. Peripheral membrane proteins, a classification based on membrane association. Definition: these proteins are temporarily associated with cell membrane. They do not penetrate the hydrophobic core of the lipid bilayer. Instead, they are loosely attached to the surface of the membrane. So, okay, so the distinction is that one particular set of lipids, oh no, not lipids, but proteins. One particular set of proteins interact directly within the cell membrane lipid chain. So it basically interacts with the hydrophobic lipid layers, like the two strands. And then there's other proteins like peripheral proteins, membrane proteins, like they're just loosely attached to the surface and need not necessarily interact with the hydrophobic part, either by interacting with the hydrophilic heads of phospholipids or by building the integral membrane proteins. Okay, how they remote, because their association is weaker, often through hydrogen bonds or ionic bonds. They can be easily detached from the membrane by changes in pH, salt concentration, or other mild treatments that don't disrupt the bilayer functions, often involved in cell signaling, acting as enzymes or regulatory subunits of integral proteins. Scaffolding, linking the members of the cytoskeleton. Scaffolding, okay. Nice verbiage, nice word. And mediating interactions between the membrane and the cell's interior. The functional words, the verb words are really interesting in your vocabulary build up. Mediating interaction is such a great rich phrase. Mediating, or like it mediates between the two layers or two positions or the two states, or facilitating, mediating, acting as a scaffolding, involved, signaling. These are like good keywords. Key differences summary feature: fibrous protein, germ protein type. Long integrated membrane proteins are varied, globular, helical, barrel shaped, often amphipathic. I don't know what this amphipathic meaning. Of a molecule, especially a protein, having both hydrophilic and hydrophobic parts. Okay, amphiphilic, ambivalent is like something, a, a type of animal that would probably operate in both water and the air. Location primarily structured outside membrane or cytoskeleton, embedded within the lipid bilayer, loosely attached to surface. Association, structural role, not direct membrane embedding. Strong permanent hydrophobic interactions with lipid. Weak temporary electrostatic, weak temporary electrostatic H bond interactions with lipid and integral proteins. Removal not relevant for membrane removal. Requires detergents to disturb bilayer. Easily removed by mild treatments like high salt, pH changes, etc. Which type of proteins are glycolipore and channel proteins? Now let's apply the integral slash peripheral classification to your examples. Glycoprotein are almost exclusively integral membrane proteins, specifically transmembrane proteins. Explanation: a glycoprotein is a protein that has carbohydrate chains, glycans, covalently attached to its extracellular portion. For these carbohydrate chains to be on the outer surface of the cell membrane, for cell recognition and adhesion, the protein part must be anchored within and often span across lipid bilayer, while the carbohydrate portion is hydrophilic. The protein portion that embeds in the membrane is hydrophobic. So the protein part that like sets its tooth within the lipid bilayer is hydrophobic. Higher inflection. Cool. Pore proteins. Pore proteins are integral membrane proteins, typically Transmembrane proteins. Explanation: pore proteins like prions, especially found in the outer membranes of bacteria, mitochondria, chloroplasts, form large non-specific channels or pores through the membrane to create a stable hole that allows molecules to pass through the hydrophobic interior of the membrane. The protein itself must be deeply embedded within and span the entire lipid bilayer. They're often characterized by a beta barrel structure that forms aqueous channel. Okay. Channel protein type. Channel proteins are integral membrane proteins, specifically transmembrane proteins. Explanation: similar to pore proteins, channel proteins form a hydrophilic pathway channel through the hydrophobic lipid bilayer. They are highly specified, allowing only certain ions or small molecules to pass. For them to create a functional tunnel through the membrane, they must be embedded in and span the entire membrane, with their hydrophobic regions interacting with lipid tails and hydrophilic regions forming the central pore. In summary, for any protein to provide a conduit or a stable anchor through the hydrophobic interior of the cell membrane, it must be an integral and very transmembrane protein. Perhaps peripheral proteins by definition stay on the surface and don't create such passage. So I am totally at lost about uh, these are the like pores are like, okay, letting things in. Because this is pores, these are like embedded very deep within. That is probably the gist. Other integral proteins have several channel proteins and selectively transport ions. Okay, they're doing it. Globular proteins, which are peripheral associated with surface of the cell, may be enzymes. Or glycoproteins, proteins with carbohydrate associations that identify the cell. Okay, so glycoproteins help identify, and more integral proteins like let molecules pass through. Got it. Cell membranes. Cholesterol molecules are embedded in animal cell membranes but not in plant cell membranes. They help make membranes along with the phospholipids impermeable to water soluble substances. Cholesterol also stabilizes the membrane. Cholesterol is added to cell membrane. Cool. Question 1 of 10. Drag the term to the graphic. Now that you have reviewed the structure of cell membrane, it's your turn to identify and build your own cell membrane. Okay, let's do it. Phospholipid. This is phospholipid. Correct. Fibrous protein. Glycoprotein. This is a glycoprotein. Pore protein. Channel protein. Which one is pore protein? Which one is channel protein? This is a pore protein. This is a pore protein, like it's pouring, and this is a channel protein. It's like letting things. I have to know the distinction between pore proteins and channel proteins. Gotcha. Select the part that forms the basic frame of the cell membrane. Select the hydrophilic head. Select the hydrophilic tails. Okay. Select the fibrous receptor proteins to replace in the pore proteins. This is like a pore protein. Okay, it's pore protein. Next. Okay, what does pore protein do? I have to like channel protein. Here you go, channel glycoprotein. This is a glycoprotein. What is the function of glycoproteins molecules in the cell? Ion transportation, no. Active transport, no. Receptor molecules, yes. Identification and identification and recognition, yes. Correct. Identification recognition is correct. Receptor molecule. What is a receptor molecules? Like other things can like sit on it. Is it that? What molecules are found in animal and cell membrane but not in plant cholesterol? Starch probably no. Starch is definitely found in animal plant cells. Please close the window. This was good. This was not unhelpful. Brain anatomy, construction of cell membrane. I think I just did this. Review study. I think I did this, right? I think I did this. There are many other stuff I can do that are similar in nature, and I would be like, especially in the computer science. Like, what is computer science? Computer programming, foundational IT skills, IT database, IT networking, IT support. Like, sure, this is a good website. Hell yeah. This is a great website. Not just good. Okay, time for some more reading on this. What type of proteins are glycoprotein? Channel proteins. Okay. What is the distinction between pore and channel proteins? This one I think would clarify that, right? Glycoproteins are almost exclusively integral membrane proteins, specifically transmembrane proteins. A glycoprotein is a protein that has carbohydrate chains covalently attached to its extracellular portions. Okay, I have read this. Pore proteins are integral membrane proteins, typically dense membrane proteins, like prions, especially found in outer membrane of bacteria. Mitochondria are large non-specific channels or pores to membrane to create stable hole that allows. Okay, stable. So this is I think the core distinction, because it was like letting molecules pass through without any barrier. And channel proteins are specific pore proteins. They are highly specific. I think this is a distinction because it like as one exits, the entryway is like blocked, allowing only certain ions or small molecules to pass. For them to create a functional tunnel through the membrane, they must be embedded in and span the entire membrane with their hydrophobic regions and interacting with lipid tails and their hydrophilic regions forming the central pore. In summary, for any protein to provide a conduit or stable anchor through the hydrophobic region, excuse me. In summary, for any protein to provide a conduit or a stable anchor through the hydrophobic interior of the cell membrane, it must be an integral and usually transmembrane protein. Peripheral proteins, by definition, stay on the surface and don't create such passage. Oh boy, this sounds so fun to read. Should I do it? Let's do it. Proteins are incredibly versatile molecule, and in the cell membrane they perform a wide array of functions that are crucial for the cell's survival and interaction with the environment. They essentially act as workers for the membrane, handling tasks that the simple phospholipid bilayers cannot. That is a good contextualizing. Like, okay, these are the workers, and the poor phospholipid bilayer chain. It cannot handle the big stuff. Transport. This is one of the most vital roles. The phospholipid bilayer is a barrier to many molecules, especially large polar or charged ones. What is the polar one? I think I remember bipolar having one charge and having like two separate charges at two separate heads. Membrane proteins overcome this by facilitating their movement across the membrane. Channel proteins, what they do, they form hydrophilic, water-filled pores or channels through the membrane. Think of it like, think of them like tunnels. Yes, channel. Channel proteins, are they like tunnels or which one? What am I thinking? There was like pore, there was like channel. Pore basically has no barrier. It's like easy entry. When open, they allow specific ions like this, or small molecules to pass through rapidly, following their concentration gradient from an area of high concentration to low concentration. This form of facilitated diffusion. This is a form of facilitated diffusion and doesn't require direct energy. Okay, energy is going to be a big part, and diffusion also. The textbook is going to talk about diffusion. Ion channels crucial for nerve impulses, action potentials in neurons, and muscle contraction. They can be gated, open, or closed in response to signal like voltage change or a binding of a chemical ligand. Okay, ion is going to be something that we are going to delve into. So, a neuroscience membrane potential. So, these are things that we can look into. I think the next portion of this series is just going to be focusing on that, right? It has to be a better uh, aquaporins, specifically designed to allow water molecules to pass through quickly, vital for maintaining water balance. Carrier proteins, transporters permeases. What they do, they bind to specific molecules on one side of the membrane and then undergo a confrontational shape change to shuttle the molecule across the other side. Right? How they work, facilitate diffusion. Like channel proteins, they move molecules down their concentration gradient, but they do so by binding and changing shape. Oh yes, this is the I thought this was like the channel protein and not the other one. The other one, channel protein. Wait, what? Yeah, I thought this was the channel protein, and this I think should be pore proteins. Am I getting it wrong? Active transport. Unlike facilitated diffusion, active transport requires energy, often ATP hydrolysis, to move molecules against their concentration gradient from low to high concentration. What was the other one doing? Concentration gradient from area of high concentration to low concentration. This is like diffusion, it happens spontaneously, naturally. And this is like going the other way. This is going in reverse and requires ATP energy kick. This is essential for maintaining specific ion concentration inside of the cell, absorbing nutrients and expelling waste. Examples: glucose transporters, GLUTs, facilitate glucose entry into cells. Sodium potassium pump, ATP phase. A classical example of active transport, pumping sodium out and potassium, K I think is potassium, right? And potassium into the cell, crucial for nerve signaling and maintaining cell volume. Okay, nerve signaling is associated with this and maintaining cell volume. Cool. Receptor function, signal transduction. What they do, these proteins act as specific antenna. Okay, this is like doing this is actually a good read now that I see this one, because it is clarifying my confusion about the different functions. For instance, what is a receptor? This is going to uh, put that tension at ease. For the cell, binding the chemical messengers like ligands From the extracellular environment, this binding triggers a series of events inside the cell, allowing the cell to respond to external signals. Okay. How they work: when a specific ligand, like hormone, neurotransmitter, or growth factor, binds to its corresponding receptor protein on the cell surface, it causes a change in the receptor shape. Yeah, the receptor is just going to change. Like, what is a receptor? I think glycoproteins are sort of a receptor. They serve as a receptor. Am I wrong? This confrontational change in their antennas, this confrontational change then initiates a cascade of intercellular events, signal transduction pathway, leading to a specific cellular response, gene activation, enzyme activation, cell division, and changes in metabolism. Examples: insulin receptor binds insulin to regulate glucose uptake. Neurotransmitter receptor bind neurotransmitter like acetylchloro acetylcholine or acetylcholine acetylcholine or dopamine, crucial for nerve communication. More about this later. Hormone receptors binds various hormones, influencing a wide range of psychological processes. Okay. Enzyme activity, what they do: some membrane proteins function as enzymes, catalyzing specific biochemical reactions directly on or within the membrane. Some membrane protein functions as enzymes, catalyzing specific biochemical reaction directly on or within the membrane. How they work: the active side of the enzyme may face either the extracellular or intercellular side of the membrane, or it may be embedded within the membrane itself. These enzymes can be involved in various metabolic pathways, signals transduction, and the synthesis of membrane components. Example is adenylyl silase, an enzyme often associated with G protein coupled receptors. It converts ATP, a crucial second messenger in many signaling pathways. I don't even want to. Digestive enzyme in the lining of small intestine, some membrane bound enzymes help break down complex carbohydrates into simpler sugars or absorptions. Okay. Cell to cell recognition, what they do: many membrane proteins have carbohydrate chains attached. This is like glycoproteins. Carbohydrate tracks act as an identifier. Okay, receptors did so without uh, glycoproteins weren't mentioned in receptors. Glycoproteins are only coming into picture when we are talking about cell to cell recognition. How they work: the unique patterns of carbohydrates on these glycoproteins and glycolipids allow cells to recognize each other. This is crucial for immune response, tissue formation, guiding cells to properly organize into tissues and organs during development. Blood typing: the AB and O blood groups are determined by specific carbohydrate change on the red blood cell membranes. Okay, AB and O. What about which one is missing? AB positive, like AB combined. Intercellular joining, cell adhesion. Some membrane proteins help cells stick to each other, forming strong connections that are essential for building and maintaining tissues. How they work: these proteins are often cell adhesion molecules, CAMs, can bind to other CAMs on adjacent cells or to components of the extracellular matrix, the network of molecules outside cell. Examples: cadherins, intergens, lectins. Do I have to Do I have to read this? I don't think I'm going to. Attachment to the cytoskeleton or extracellular matrix ECM. Membrane proteins that can anchor the cell's internal scaffolding to the plasma membrane and also connect the membrane to extracellular. How they work? Structure support help maintain cell shape and allow cells to move and interact with the environment. They also enable outside in and inside out signaling, where changes in ECM can influence cell behavior. Changes inside the cell can affect the its addition to the ECM. Examples: intergenes, as mentioned above, are key players in this function, bridging the gap between the internal skeleton and the external ECM, which is cytoskeleton and extracellular matrix. In summary, membrane proteins are dynamic and essential components of the cell membrane, transforming it from a simple boundary into a highly active and interactive interface that mediates virtually all communication and exchanges between the cell and its surroundings. Whew, sheesh. How much time has been? Can I see? Okay, it's been 30 minutes. I can go for another 30 or so minutes. That would be the worst thing in the world, would it? We have two books we probably should check out. Ion channels. This is going to talk about let me pause and check the progress on the. And I am back. So, we were trying to see this book. So, this is the book Anatomy and Physiology. And this is quite an extensive book. So, I just went to chapter 17, cardiovascular system, the heart. And well, as you would have it like this, plenty of stuff to read. Plenty of stuff to read. And it would be interesting for me to follow this chain of lectures to hopefully eventually get to that position. How much time is this going to take me though? I think this entire endeavor is going to take me let me see, 60 videos, right? So 60 weeks. 60 week is almost in a year, more than a year, 52 weeks a year. So 60 days, if I could like watch one video per day, which should be seemingly difficult with the full-time or almost full-time job endeavor that I have in front of me. So maybe within the next 3 months, June, July, August, maybe I can try and pull like 10 of them. 10 of them would be like good. 10 of them wouldn't be the worst thing in the world, watching 10 videos. So maybe my internal target should be at least 10 of the videos, and once I'm done with the workload, maybe within the next, I don't know, 50 or 2 months, 2 or 3 months, watch the rest of the stuff and read through rest of the stuff, right? That would be amazing. But in any case, this is the, like, again, this is like an extensive read, this book, if I want to read. And this is like chapter 3. It deals with organization at a cellular level. So introduction to study of cells at a basic unit of life. 3.2 is cell membrane and fluid mosaic model. We are dealing with the fluid mosaic model right now, as of now. Components of plasma membrane. This I don't know what this is, like components of plasma membrane. Let's read. The plasma membrane protects the cell from external environments, mediates cellular transportation, and transmits cellular signals. Okay. Describe the function and components of plasma membranes. To primary, one is the protect cell from surroundings, composed of phospholipid bilayers, extracellular matrix, and other cells to help group cells together from the tissue. The membrane also maintains a cell potential. In short, the cell is represented by a castle. The plasma membrane is the wall that provides structure for the building. Okay, I understand the analogy. And this is like the structure: extracellular fluid outside, nucleus in inside, cytoplasm in the inside, cell membrane. I think would recognize this right now. Carbohydrate, glycoprotein, basically glycoprotein, and it's like carbohydrates like sitting directly. Okay, didn't know that. Globular proteins, this is like globular proteins, protein channels transport cholesterol, glyco lipids. These are like direct glycolipids. Okay, surface proteins, globular proteins. Globular proteins are really like, as opposed to protein channels. No, protein channels are also letting things in, but globular proteins are like, it probably takes energy, ATP, to get through this thing probably. Filaments of cytoskeleton. Okay, this like, uh, what noodles like, dry noodles like structure. Alpha helix protein, integral protein, peripheral protein. Alpha helix protein is this one. Okay, and peripheral protein is this one. Okay, phospholipid bilayer, phosphatidylcholine, choline. Hydrophobic head, hydrophilic head, excuse me. The plasma membrane is composed of phospholipids and proteins that provide a barrier between the external environment and the cell, regulate the transportation of molecules across the membrane, and communicate with other cells via protein receptors. Okay, the plasma membrane and cellular transport. The movement of substance across the selective permeable plasma membrane can be either passive, that is occurring without the input of cellular energy, or active, i.e., transport requires the cell to expend energy. The cell employs a number of transport mechanisms that involve biological membranes. Number one, passive osmosis and diffusion. Transport gases such as O2, O3, O2 and CO2, and other molecules of ions, transmembrane protein channels, transmembrane protein channels and transporters transport small organic molecules such as sugar and amino acids. Endocytosis transports large molecules or even whole cells by engulfing them. Endocytosis. Exocytosis removes or secretes substances such as hormones and enzymes. I don't know what these terms mean. Plasma membrane and cellular signaling. Among the most sophisticated functions, the plasma membrane is among the most sophisticated functions of the plasma membrane is its ability to transmit signals via complex proteins. These proteins can be receptors, which work as receivers of extracellular inputs, as an activators of intracellular process. Okay, this is a key line. Which works as receivers of extracellular inputs and as activators of intracellular process, or makers, which allow cells to recognize each other. Membrane receptors provide extracellular attachment sites for efforts like hormones and growth factors. Effectors like effectors. The receptors basically provide extracellular attachment sites for effectors like hormones and growth factors, which then trigger intercellular response. Some viruses, such as human immunodeficiency virus (HIV), can hijack these receptors to gain entry into the cell and causing infection. Membrane markers allow cells to recognize one another, which is vital for cellular signaling process and processes that influence tissue and organ formation during early developments. This marking foundation also plays a later role in the self versus non-self distinction of the immune response. Marker proteins on human red blood cells, for example, determine blood type. So what I get is like there's receptors. They receive things such as hormones and enzymes, and then there's like just markers like the glycoproteins basically serve as markers is my idea. Okay, it wasn't that much of a scary read. Cell signaling, let's go. Wow, there's nothing here. Great. Types of receptors, let's see. Intracellular receptors are located in the cytoplasm of the cell, and intercellular receptors are basically receptors that will be found inside the cell, inside layer of the cell. Ligand molecules, I still don't know what that is, that can pass through the plasma membrane. Cell surface receptors bind to an external ligand molecule and convert an extracellular signal into an intracellular signal. Three general categories of cell surface receptors include ion channel, G proteins, and enzyme linked proteins receptors. Okay, there are like three. That is a good information. Three general categories of cell surface receptors include ion channel, G proteins, and enzymes. Ion channel linked receptors bind a ligand and open ion channel. Ion channel linked receptors bind a ligand and open a channel through membrane that allows specific ions to pass. So it wouldn't necessarily need active energy, would it? G protein linked receptors bind a ligand and active a membrane protein called G protein, which then interacts with either an ion channel or an enzyme in the membrane. Which then interacts with either an ion channel or an enzyme in the membrane. Bind a ligand and activate a membrane protein called G protein. I have to know more about this. Enzyme linked receptors are cell surface receptors with intracellular domains. Enzyme linked receptors are cell surface receptors that with intracellular domains that are associated with an enzyme linked. Okay. Integrated protein, a protein molecule or assembly of proteins that is permanently attached to a biological membrane. Transcription, a synthesis of RNA under the direction of DNA. Still failing to realize why this one was like crucial. Why was it included? Bacteria that are pathogenic to humans can release poisons that interrupt specific G protein linked receptor function, leading to illness. In cholera, the waterborne bacteria produces a toxin, chlorogen, that binds the cells lining in a small intestine. This toxin then enters these intestinal cells, where it modifies a G protein that controls the opening of chloride channels and causes it to remain continuously active, resulting in large losses of fluid from body that can lead to potentially fatal dehydration. Receptors are protein molecules in the target cell or on its surface that binds ligands. There are two types of receptors: internal receptors and cell surface receptors. Internal receptors, and by the way, this is a chapter about receptors. Internal receptors, also known as intracellular or cytoplasmic receptors, are found in the cytoplasm of the cell and respond to hydrophobic ligand molecules that are able to travel across the plasma membrane. Once inside the cell, many of these molecules bind to proteins that act as regulators of mRNA synthesis. This is where I am losing the plot. Once inside the cell, many of these molecules bind to proteins that act as regulators of mRNA synthesis to mediate gene expression. Okay, so this like proteins are going through some mRNA and DNA channels that I read in HSC, but I'm totally forgetting what that was. Gene expression is the cellular process of transforming information in its cell's DNA into sequence of amino acids that ultimately forms protein. Again, this portion I have to be visualized and I have to be lectured. And I think Professor Fink has a playlist on biology that I need to get into to fully understand like what is happening here. I think he's the only person that can simplify it for me. There are people who can simplify it for me, but he has this unique ability to um, communicate information in a matter that would stick to you. Like you'd follow it and you'd find it interesting at the same time. When the ligand binds to the internal receptor, a confrontational change exposes a DNA binding site to the protein. The ligand receptor complex moves into the muscle, it moves into the nucleus, binds the specific regulatory regions of the um, chromosomal DNA, and promotes the initiation or of transcription. So this is like protein transcription or DNA transcription, whatever. Internal receptors can directly influence gene expression without having to pass a signal on the other receptors or messengers. Okay, internal receptors have to. Thanks. Internal receptors bio. Receptor signals transduction and so I don't want to play music. This video is part of what? This video is part of what series actually? Is this part of biology or what? Biochemistry. Okay, introduction to biochemistry, amino acids, protein structures, enzymes, nature's factory workers, carbohydrates part 1, simple sugars and Fischer projections, carbohydrates part 2, polysaccharides, lipid part 1, TGA fatty acid and terpenes, lipid part 2, steroids, structure of the cell membrane, active and passive transport. Yes. Receptors, signals, transduction, high phosphorylation cascade, nucleic acid, DNA and RNA, DNA replication, copying the molecules of life, transcription and translation, forming DNA to protein, transcription and translation. Transcription is the word, the term that I realized from this. Mechanisms of DNA damage and repair, metabolism and ATP, cellular respiration part 1, glycolysis, cellular respiration part 2, citric acid, cellular respiration part 3, electron transport chain, photosynthesis, cell communication, what is cancer, illicit drugs, how do they work, pharmaceutical drugs, inhibitors, and nature of diseases, visualizing DNA in virtual reality VR. Okay, I'm going to watch this. There's also anatomy and physiology course that I can watch, AP chemistry. There's linear immunology and general chemistry. So this courses should probably go hand in hand. There's one on biology, bio biology slash genetics, American history, and geology, organic chemistry. Okay, I think I'm going to watch at least some of them, and if I watch these lectures, it's not going to be to comment on them or make study sessions on them. It's just going to be for private consumption on a smaller device to learn more about it. Anywho, I have found my thing. Let's move on. Cell surface receptors, also known as transmembrane receptors, are cell surface membrane anchored or integral proteins that bind to external ligand and ligand molecules. This type of receptors spans the plasma membrane and performs signal transduction, converting an extracellular signal into an extracellular signal into an intracellular signal. Ligands that interact with cell surface receptors do not have to enter the cell that they affect. Wait, what? Ligands that interact with cell surface receptors don't have to enter the cell they affect. Cell surface receptors are also called cell specific proteins or markers because they are specific to individual types of cells. Okay. What is the distinction between internal receptors? Internal receptors, things can just pass through them, hopefully. That is what cell surface receptors are. Things are not passing directly onto the inner core of the cell, but can have an effect. For instance, I think hormones and enzyme stuff like that. Ligands that interact with cell surface receptors do not have to enter the cell that they affect. Cell surface receptors are also called cell specific proteins or markers because they are specific to individual cell types. Each cell surface receptor has three main components: an external ligand binding domain, extracellular domain, a hydrophobic membrane spanning region, and an intracellular domain inside the cell. The size and extent of each of these domains vary widely depending on the type of receptor. So let's try to see ions opening channel. So they look like they need enzymes, like they need ATP to open up, right? Oh no, this is like different things. Surface cell receptors are like things that stick out. For instance, if this is like the outer layer, like they are like here, and this is outer layer, this is inner layer, these things would be like here, like here. Ion channel linked receptors. What are the three categories? Ion channel, okay, protein enzyme linked receptors. Internal receptors, cell surface receptors, ion channel, G protein, enzyme linked receptors. Okay, so again, they are like not making it easy for us to them, are they? They're having basically two separate definitions, right? One is like where the protein molecules, like where the protein is with respect to the layers of the membrane, like the membrane itself, like is it outside or is it inside, or is it like throughout. And the other one is kind of a functional based, which is like ion linked receptors. Ion channel linked receptors bind a ligand and open a channel through the membrane that allows specific ions to pass through to form a channel. This type of cell surface receptors has an extensive membrane spanning region in order to interact with the phospholipid fatty acids that form the center of the plasma membrane. And many of the amino acids in the membrane spanning region are hydrophobic in nature. Conversely, the amino acids that line the inside of the channel are hydrophilic to allow the passage of water or ions. When a ligand binds the extracellular region to the channel, there is a confrontational change in the protein structure that allows ions such as sodium, calcium, magnesium, and hydrogen to pass through. I think I'm going to leave it up to more visualizing element, like I have to search YouTube for this. form a port through plasma membrane that opens when signaling molecule binds. The open port that allows ions to flow into or out of the cell. So it doesn't require any ion, basically because they're ion charged, that doesn't require any particular uh, work to be done. It's like pure diffusion. Protein linked receptors. G protein linked receptors bind a ligand and activate a membrane protein called G protein. The activated G protein then interacts with either an ion channel or an enzyme in the membrane. All G protein linked receptors have several transmembrane domains, but each receptor has its own specific extracellular domain and G membrane binding site. Each receptor has extracellular domain specific to it. Okay, cell signaling using G protein linked receptors occur as a cyclic series of events before the ligand binds. The interactive G protein can bind to a newly revealed site on the receptor specific for its binding. Once the G protein binds to the receptor, resultant shape change activates the G protein, which um, releases GDP to pick up GTP. The subunits of G protein then split into the A subunit and B subunit. And okay, this is too complex. I think I'm going to have to rely on YouTube visualization as well. Enzyme link receptors are cells of receptors. Domains are associated with enzyme. It means in some cases the internal domain of the receptor is an enzyme. Link receptors. Okay, I am not going to get more details on this. This is like it. I was studying what? I was studying signals, type of receptors, G.4a. Cell signaling and type of receptors. Transport across membranes. Let's just check this one out. Holy hell, this has its own like diffusion, tonicity, role of passive transport, electrochemical gradient, content of next session, endocytosis, primary active transport, secondary active transport. I'm not going to get into that now. Receptors are basically like they're not transporting. That's why there was no particular molecules going in, particular molecules going out, because receptors are basically receiving, and something is basically sits on it, and like it translates the signal that we get from the outside into something that the cell should do in the inside. Nucleus and ribosome, okay, organelles, plasma membrane and the cytoplasm, endoplasmic reticulum, the Golgi apparatus, lysosome, peroxisomes, mitochondria, microfilaments and cytoskeleton, external cellular components. Okay, I'll have to keep the tabs open because I'm going to link this to certain things. This one looks too complex, by the way. Dr. Kevin Ahern and Dr. Indira Rajgopal's electronic book is aimed at helping students learn the basics of biochemistry in a fun and engaging format. So maybe Professor Dave's lecture series should coincide with this one. I'll link this just in case, putting it all together. Black matter is metabolism 1, metabolism 2. He was dealing with respiration, maybe metabolism as well. Cells, water and buffers. Okay, we'll deal with this, not into this. Lastly, we are here. Let's see how much time has spent. If the video has surpassed like the 1 hour mark, I think I'm going to pause, or even like 50-minute mark, I'm going to pause and maybe come back to this afterwards, or maybe miss this altogether. I think we're in a state where we might just miss this altogether, because I need additional information right now. The most I can do is just read this on, and I think I'd be better off if I gathered additional information, insights into the Professor Dave series on biochemistry, and basically read some, not read but watched some Professor Fink lectures on biology, basic biology just, and some of his next lectures, and I think I should be better off reading up some secondary, like college level biology that I had, the book that I had, just to get a basic understanding of the cells. Maybe then I should come back to this, maybe then, but I will try to link this to the video, part 2 video that this is. And I think I'm going to call it a day now.